Hello students, welcome once again to our class of BCH 408. Today we are on lecture number 3. Okay. Now let's look at the summary of lecture 2 before we can proceed to what we have today as a reminder of what we did previously. In that class, lecture 2, we defined heme, its chemical structure and biological significance. We identified the major hemoproteins in the body, described the sites and cellular locations of heme synthesis. We went ahead to outline the steps and the key enzymes involved in heme biosynthesis. We explained the mechanism regulating enzyme synthesis. In today's topic, we are going to deal with chemistry of oxygen and carbon 4 oxide transport. Chemistry of oxygen and carbon 4 oxide transport. What will be our learning objective for this lecture? In other words, at the end of this lecture, what will be your take home? You should be able to define the roles of oxygen and carbon 4 oxide in biological system, describe the chemical properties and of oxygen and carbon 4 oxide, explain the different mechanism of oxygen transport, understand oxygen dissociation curve and factors affecting oxygen binding and release in the heme molecule. You should be able to outline 3 major forms of carbon 4 oxide transport in blood. In addition, you should be able to explain the role of carbonic anhydrase and enzyme in the conversion of carbon dioxide and bicarbonates in red blood cells. You should be able to describe what what chloride shift is and its importance in maintaining ionic balance during carbon 4 oxide transport. You should be able to differentiate between gas exchange processes occurring in the lungs and in the body tissues, as well as be in a position excuse me, as well as be in a position to discuss or describe the chemical basis for Bohr effects and Haldane effects as it relates to gaseous transport in blood. Now let's get started with the roles of oxygen and carbon 4 oxide in blood in biological system. Let's first of all start by looking at the overview of respiration. Respiration is a set of biochemical and physical processes by which living organisms obtain energy from nutrients. It involves intake of oxygen and release of carbon 4 oxide. It occurs at 2 levels: at the level of breathing, which is gaseous exchange, that is at the cellular levels, and the physiological level. The physiological level involves breathing and gas exchange, while the cellular level involves the production of energy. Now, there are two types of respiration: anaerobic respiration, which requires oxygen and produces ATP, and anaerobic respiration, which occurs without oxygen and produces less ATP. Now, that is by way of the overview of respiration. Now, let's look at external and internal respiration. External respiration is the process by which exchange of gases occur between external environment and the blood. That is, external environment and the blood, looking at them now as two different compartments. Internal respiration, on the other hand, is the exchange of gases between the blood and body tissues. In summary, external respiration prepares oxygen for transport, while internal respiration ensures cellular utilization of oxygen. We may like to know the importance of gaseous transport in living system. Gas transport is essential for maintaining life and cellular functions. The key roles involved the following. 1. It delivers oxygen to cells for energy production. It removes carbon dioxide or carbon dioxide, thereby preventing toxicity. It maintains acid-base balance, which regulates pH and supports homeostasis. Why gaseous transport is needed? Why is it needed? Oxygen is polysoluble in plasma, requiring transport by hemoglobin. On the other hand, carbon dioxide must be efficiently removed to avoid acid accumulation. Now, the role of oxygen in metabolism. Oxygen is central to energy production, particularly in aerobic organisms. It functions as an electron acceptor in cellular respiration, enables oxidative phosphorylation, and is involved in detoxification reactions, where oxygen is used to attach to molecules. Which are discussed in 407. Now, carbon dioxide in metabolism. Carbon dioxide is a metabolic waste product that plays a regulatory role, such as formation of carbon dioxide produced during the Krebs cycle, oxidative decarbonization of nutrients, among others. The physiological role include regulation of blood pH, reaction with water within the cell to form carbonic acid, maintenance of acid-base balance, control of breathing. We must emphasize that carbon dioxide level regulates respiratory rates via chemoreceptors. Now, let's look at chemical properties of carbon 4 oxide or carbon dioxide. Carbon dioxide is more soluble than oxygen in plasma. About 5 to 10% of carbon dioxide is transported, dissolved directly in blood. So you can see the difference. And of course, you know that carbon dioxide is a waste product that must be removed from the blood. Formation of carbonic acid. Carbon dioxide will react cellular water to form carbonic acid. This acid is weak and contributes to the regulation of pH in blood. Ionization to bicarbonate, which is a major form of transportation of carbon 4 oxide or carbon dioxide in blood. This transports about 70% of carbon dioxide in the blood. That is, bicarbonate, that is carbon 4 oxide or carbon dioxide, is transported as bicarbonate in blood to the tune of 70%. This reaction is catalyzed by carbonic anhydrase, and the reaction is reversible, which means carbonic anhydrase can form carbonic acid by reacting water with carbon dioxide, and it can also dissociate it by forming bicarbonate and hydrogen ion. Excuse me, please. Carbon dioxide also have the ability to form carbamino compounds. They have the ability to form carbamino compounds by reacting with amino group in proteins, especially proteins of hemoglobin. In that way, they are also transported, and that kind of transportation of carbon 4 oxide account for about 20 to 25%. Carbon dioxide plays a central role in bicarbonate buffer system. When you have increased carbohydrates, of course, it means, sorry, when you have increased carbon dioxide, it means you have increased hydrogen ion. And of course, that will lead to decreased pH. If you have decreased carbon 4 oxide or carbon dioxide, you have increased pH. That is, it makes the blood becomes alkaline. Why increased pH? That increase hydrogen ion and decrease pH make the blood become acidic. So carbon dioxide could be responsible for acidosis or alkalosis. It can also play a role. Carbon dioxide can also play a role in determining the partial pressure of gases in the blood. As carbon dioxide transport is driven by partial pressure, high partial pressure in tissues will lead to diffusion into blood. That is, when you have much partial pressure of carbon dioxide in tissues, you will have more carbon dioxide diffusing into the blood. What it means by partial pressure is when you have, the gases are made up of mixture, the air is made up of a mixture of gases. So individual gas contribution constitute the partial pressure of that gas. So if that of carbon dioxide is more, then it means that more carbon dioxide will diffuse into the blood. If it is low in the lungs, it means it will lead to diffusion out of the blood. Remember, the high partial pressure leads to diffusion into the blood, then low partial pressure will lead to diffusion out of the blood. So it's a case of in and out. Now let's look at the mechanism of transport. I try to simplify it here. You see, from the picture on the screen before you, you can see the red blood cell. You see the hemoglobin molecule, and you see the oxygen molecules. Now, as they bind to the hemoglobin molecule, they become tightly bound with the hemoglobin within the red blood cells. Now, the red blood cells is taken to the tissues where the oxygen is released. This mechanism can be explained. Let us look at the mechanism by which the heme present in hemoglobin transport oxygen. Now, the heme group in the hemoglobin molecule is the central core or the center of activity of that molecule with respect to oxygen transport. How does this play out? Now, the oxygen molecule attaches to the central ion that is present in the heme group. That binding creates 6 coordination bond. That is, one from oxygen, one from the globin, histidine, protein, and 4 from the porphyrin of hydrogen atom. Now let's talk a little about the porphyrin ring. The porphyrin ring actually has 4 nitrogen atoms. Now these hydrogen atoms are the ones that actually hold the ion in the center to provide a stable bonding platform. Now it is this ion, that is ion 2, ferrous ion, that binds directly to the oxygen. And of course, this binding is reversible. It enables oxygen to load and to release and to offload when necessary. Now, the globin protein comes from the histidine and attaches to the ion from below, thereby stabilizing the heme structure. So with this mechanism of binding, the heme group present in the hemoglobin is able to transport oxygen. How about dissolved oxygen in plasma? Now, I told you before that oxygen is parallelly dissolved in plasma. The amount of the gas dissolved in a liquid is directly proportional to its partial pressure. And of course, that is what the Henry law states that the concentration of gas in liquid is proportional to its partial pressure. According to this equation you are seeing here, where C stands for concentration of dissolved gas, K solubility constant, and P the gas partial pressure. So the higher the gas, the higher the pressure, the more the gas, and the lower the pressure, the less the gas in solution. Only about 2% of total hydrogen is transported dissolved in plasma. This is because oxygen is not polar, and therefore the interaction with water is low. Remember, at the beginning of this lecture, I told you that plasma is mainly water. So because of that, and being that oxygen is not polar, it is poorly dissolved. So what could be the major transport form, therefore? About 99% of oxygen is transported bound to hemoglobin as oxyhemoglobin because of the advantage provided by the ion or the heme group. Therefore, the body relies heavily on hemoglobin to increase oxygen supply to its tissues. Let's look at the oxygen dissociation curve. As you see before you, the structure here, or the graph here, represents oxygen dissociation curve. The x-axis is the partial pressure of oxygen measured in millimeter mercury, while the y-axis is the percentage saturation of hemoglobin. You can see the graph is taking a shape of S, so we refer to it as sigmoidal. Now from this graph, something is clear. You can see that for increased affinity for oxygen, there will be a decrease in temperature, decrease in partial pressure of carbon dioxide, decrease in 2,4-diphosphoglycerates, and increase in pH. For there to be a decreased affinity for oxygen, there will need to be increase in temperature, increase in partial pressure of carbon dioxide, increase in 2,3-diphosphoglycerates, and a decrease in pH. So this is what we refer to as oxygen dissociation curve, which has an S shape that flattens out at the plateau. That is the top, or level out at the top. What factors can affect oxygen binding and its release. In other words, what factors can affect oxygen binding to hemoglobin and its release from hemoglobin? Of course, as I showed, or as was seen just now in the oxygen dissociation curve, the factors include partial pressure of oxygen, partial pressure of carbon 4 oxide, blood pH, temperature, 2,3-bisphosphoglycerate, hemoglobin type, carbon monoxide presence, and cooperative binding of heme. These are the factors that can both affect oxygen binding as well as its release. We shall deal with a few of these in a few minutes. Now, we look at carbon 4 oxide transport in blood. Having examined the mechanism by which oxygen is transported, let's look at that of carbon 4 oxide or carbon dioxide. Recall, we said that carbon dioxide is more directly dissolved in blood plasma and transported according to Henry's law. Remember, Henry's law states that the more the partial pressure of the gas, the more it is dissolved in the liquid. That is, the concentration is directly proportional to its partial pressure. Secondly, carbamino compounds. About 25% of carbon dioxide bind to proteins, mainly hemoglobin. Thirdly, bicarbonate ions forms the major route for transporting carbon dioxide through the blood. So, bicarbonate forms the major form of carbon dioxide transportation in blood. This is because carbon dioxide diffuses into red blood cells where it is converted to carbonic acid by the action of carbonic anhydrase. Let's look at the role of carbonic anhydrase in blood. The roles are twofold. One, it catalyzes the reaction whereby without this enzyme, the reaction to carbonic acid will be very slow. But in the presence of this enzyme, it becomes very fast. How about the role of carbon dioxide transport? From tissues to lungs. In the body tissue, carbon dioxide produced by metabolism diffuses into the red blood cells. Here, carbonic anhydrase converts the carbon dioxide and water into carbonic acid. What role does it play in the lungs? In the pulmonary capillaries, bicarbonate ion re-enters the red blood cells, combines with hydrogen ion to form bicarbonate. Carbonic anhydrase converts bicarbonate to carbon dioxide and water. And this is where elimination occurs. The carbon dioxide is then expelled and exhaled. Now, what is the importance of this enzyme in blood? It ensures rapid gas exchange, enables efficient carbon dioxide removal, and maintains blood pH. It also supports continuous cellular metabolism. There is an important term we need to know that has to do with chloride shift and ion balance. By definition, the chloride shift simply means exchange of bicarbonate ion with chloride ion. Exchange of bicarbonate ion with chloride ion across the red cell membrane to maintain electrical neutrality during carbon dioxide transport. Now, the picture here, you can see the red blood cell with the plasma, and you can see that carbon dioxide is crossing the membrane, the red cell membrane, and to form bicarbonate. And you see here Ca. Ca is carbonic anhydrase that converts carbon dioxide and water to bicarbonate, and Ca also splitting the bicarbonate to hydrogen ion and bicarbonate ion. Furthermore, the bicarbonate ion is leaving the cell membrane, it's leaving through the cell membrane, and a chloride ion is now entering. Just to, as one ion is leaving, another one is replacing it. Because it's chloride that is leaving the outside to the inside, it is referred to as chloride shift. It helps to balance the bicarbonate ion that is leaving the cell in order not to make the inside of the cell high, more positive than it should be. Now, I have taken time. Let me take this time to explain the mechanism of chloride shift, which the diagram before this clearly does explain. Now, carbon dioxide is produced by cell. It diffuses into red blood cell. It is converted to bicarbonate according to this equation. Of course, bicarbonate accumulates inside the cell, diffuses out into the plasma. To maintain charge balance, chloride moves into RBC, into the red blood cells. This inward movement is what is referred to as chloride shift. This inward movement of chloride ion is what is referred to as chloride shift. So this is simply a repetition of what was earlier said. The emphasis is for you to understand what chloride shift means. In the lungs, however, the process is reversed. Bicarbonate ion move back into the cell. Chloride ion move out of into the plasma. Carbon dioxide is regenerated and exhaled or expelled. What transport protein is involved in all this? The protein that is involved is called anion exchanger. Anion exchanger. It facilitates chloride ion and bicarbonate ion exchange. Now, can this chloride shift be important? Yes, it can be in the following ways. A maintenance of electrical neutrality in the blood cell, efficient carbon dioxide transport, pH regulation, facilitation of continuous gas exchange, and osmotic. Now, in the lungs, gas exchange happens because of difference in concentration. The image that is shown on screen illustrates the gas exchange in the lungs, where deoxygenated blood enters. The oxygenated blood is shown by this blue arrow. It is high in carbon dioxide. This is shown also in blue. Air in the lungs is high in oxygen, that is low in carbon dioxide. Oxygen moves into the blood from the lungs. This happens by diffusion. Of course, from high concentration of high to low concentration. When the oxygen enters the red blood cell, it binds with hemoglobin, and the blood becomes rich in oxygen, or oxy. We refer to such hemoglobin as oxyhemoglobin. Now, carbon dioxide moves out of the blood into the lungs, and it is then expelled from the body. Oxygenated blood that is, blood containing or rich in hydrogen, leaves the lungs and is carried into various tissues. So this is how the body taking oxygen and transport them into various tissues, right from the lungs. In the tissues, the diagram before you is also illustrating gas exchange, showing how oxygen is delivered to the body cells and how carbon dioxide is removed. Blood flowing through the capillaries arrives rich in blood and in oxygen. Because the concentration of oxygen is higher in the blood than in the surrounding cells, oxygen diffuses out of the cell, passing through the capillary walls and enters the cell where it is used for metabolism and energy production. At the same time, the cells are actively producing carbon dioxide as waste products of metabolism. Since the concentration of carbon dioxide is high or higher in the cell than in the surrounding blood, carbon dioxide diffuses from the cell into the capillaries. The blood, now carrying more carbon dioxide and less oxygen, becomes deoxygenated and is transported back towards the lungs for gas exchange. Now, the blue spread you see inside the cell here represents blood cells that has delivered oxygen and taken up carbon dioxide. Here the capillary is red because the blood here is rich in oxygen, and the right is low in oxygen but rich in carbon dioxide after taking up the carbon dioxide that is produced during respiration, cellular respiration. Well, we now look at chemical basis for the Bohr and Harding effects. Starting with Bohr effects. It simply states that increased carbon dioxide and hydrogen ion concentration reduce the affinity for hemoglobin, reduce the affinity of hemoglobin for oxygen, thereby promoting oxygen release in tissues. Note, the increase in carbon dioxide and hydrogen ion concentration reduce the affinity of hemoglobin for oxygen, thereby promoting oxygen release in tissues. That is by way of definition. Now, what is the chemical basis? You can see from the formation of hydrogen ion, which I have mentioned to you before, to the protonation of hemoglobin and reduced oxygen affinity. So to understand this, you simply follow the formation of hydrogen ions, protonation of hemoglobin, and reduced oxygen affinity. Of course, oxygen affinity will be reduced once carbon dioxide concentration is increased. How about Haldane effect? It describes how oxygenation of hemoglobin reduces the ability of the molecule to carry carbon dioxide, while the oxygenation enhances carbon dioxide transport. Now, let me take that again. The Haldane effect describes how oxygenation of hemoglobin reduces its ability to carry carbon dioxide, while deoxygenation enhances carbon dioxide transport. Now let's look at the chemical basis for that statement. Just like the Bose effect, we have oxygenation reducing binding of carbon dioxide. We also have release of hydrogen ions according to the equation I had earlier described that leads to the expulsion of carbon dioxide. Now, for you to understand this very well, simply go to the earlier slide, and look at the pathway of how carbon dioxide and hydrogen ion combine to form carbonic acid, and how they split to form carbon dioxide and bicarbonate ion and carbon and hydrogen ion. Carbamino compound breakdown. Carbon dioxide bound to hemoglobin to form carbamino hemoglobin, and it's released when oxygen binds. In tissues, it's a reverse process. Deoxygenation or deoxygenated hemoglobin bind carbon dioxide more readily, while buffered ion effectively enhances carbon dioxide uptake. Okay, students, we have come this far. I have listed out a set of 5 questions that you can use to appraise yourself, your level of understanding of what has been presented in this video lecture. Wherever you have difficulty, I encourage you to communicate among yourselves, and like I said in the last video, I will fix a time by which we can physically meet to interact and discuss these questions. Until then, it is bye from me. Thank you.